question that chapter 13 starts to answer is the genetic material of organisms made of DNA, or is it the protein? Since a chromosome is DNA with tightly bound proteins, scientists back in the day weren't sure which part of the chromosome was responsible for heredity. One of the first scientists to lead to the discovery that DNA is responsible for genetic information is Frederick Griffith in the 1920s. So his experiment, he was trying to come up with a vaccine that essentially inoculated against pneumonia or prevented pneumonia in different people, since that was a problem in the 20s. So he used mice in his experiments, and what he did was he injected a mouse in a control experiment with the living S cell. So there's something that you need to know. There's two different strains of this bacteria, which bacteria causes pneumonia. So one of the strains, which is the S strain, is the serious strain. So this is the one that's pathogenic and it causes pneumonia. So when he injected the S strain into the mouse, obviously it had pneumonia and it died. The other type of strain, which is non-pathogenic, which means it does not cause the pneumonia, is a R strain. So he also injected the mouse with an R strain. And the difference between the two of them is the R strain doesn't have a capsule around the actual cell. And you might need to look up what a capsule is on a prokaryotic cell. But the R strain does not have it, and it does not cause pneumonia. So in another experiment, he heat treated. So what we know about bacteria is when you heat it to extreme temperatures, for the most part, it will die. So he heat treated these S cells, and of course, the mouse was healthy. The bacteria wasn't active in a sense, and it did not cause pneumonia. Now, in his experiment and trying to come up with this vaccine, he also did a mixture. So he mixed the heat treated or the killed S serious cell, and then he mixed it with the non-pathogenic R cell. What he found was the mouse ended up dying. So his conclusion is what transformed, what happened to make the mouse die when the S cells that were there were heat treated. It couldn't have been that. What exactly happened to the R cells to cause the mouse to die? So the conclusion by Frederick Griffith is that the living non-pathogenic R bacteria strand was able to transform into the deadly S bacteria strain by an unknown heritable substance. Now, Griffith did believe this to be DNA, but many scientists still did not believe it. So 3 additional scientists, or bacteriologists, are Avery, McCarty, and McLeod. So what they did was they further tested the heat-killed S strain, the pathogenic strain of bacteria, and they removed proteins, they removed polysaccharides, they removed the coats. They figured out that the harmless bacteria still had the ability to cause disease. And through a series of experiments, they concluded that this transforming agent that was responsible was, in fact, DNA. Another experiment that was additional evidence that DNA is responsible for genetics is an experiment done by Hershey and Chase in the 1950s. They used viruses in their study to try to figure out if DNA or protein still is responsible for transformation and genetics. Something you need to know about viruses, essentially all a virus is DNA coated with protein, and protein being the protective coat on the outside. Now, a virus attaches to another cell and it injects its information, which is DNA, but they didn't know this at the time, into the cell, and reprograms the cell, and the cell, the host cell, is able to make copies not of itself, but instead of the virus. And then eventually it gets to a point where it explodes, and then the copied viruses are released into the organism and infects that organism, or kills the bacteria if it's a bacteria cell. So back to Hershey and Chase, they studied bacteriophages. Now, these are specific types of specific types of viruses that infect bacteria. And what they did was they substituted a element within the structure, which is working like a tag. So on certain types of viruses, they swapped out a normal sulfur atom with a radioactive sulfur on the protein layering. In a different bacteriophage, they swapped out a phosphorus atom and made it essentially radioactive. So the goal was to study the contents after the virus infected the bacteria cell. So in this diagram, you can see two different experiments taking place. Along the top, we have the radioactive sulfur, which is embedded in the protein of that bacteriophage, which again is a virus. So the virus attaches to the DNA, and it is centrifuged to blend it basically and separate it to see where is the radioactivity. At the end of the experiment, it was shown that the radioactivity is still in the liquid, not in the pellet at the very bottom. The pellet is actually the bacteria. So there is no radioactivity in the bacteria itself, which indicates that the radioactive protein is not being injected within that bacteria cell. So in the second experiment down at the bottom, you can see that now there's radioactive phosphorus within that bacteriophage. The same experiment is performed and the radioactivity is now within the pellet, indicating that that bacteriophage injected its DNA, not protein, into the bacteria. Now that it was discovered that DNA is in fact the genetic information, now scientists need to figure out what exactly is the structure of DNA. One of the scientists to study DNA is Edwin Chargaff, and he figured out two important rules about DNA. Before we get into the rules, something that was already known about DNA is that it consisted of three parts. One part is a nitrogenous base, another part is a pentose 5-carbon sugar known as deoxyribose, another part of a nucleotide, which again, DNA is a polymer of nucleotides. The last part is a phosphate group. So those three parts make up DNA, repeating units to make up DNA. Now, something that he figured out in studying different types of organisms, different types of species, is that the DNA composition varied between different species. So a sea urchin had roughly 33% DNA composition, whereas a bacteria cell would have somewhere around 24%. So there was a difference in how much DNA different organisms had in their cells. So that was something that wasn't widely known. The other thing that he figured out was something known as Chargaff's. One of the more controversial topics in science and in biology is who should get credit for the discovery of the double helix that is DNA. So it's thought widely that Rosalind Franklin, she should have received the Nobel Prize, although it was passed when she was deceased, which does not make her a candidate, but that she, in fact, before she passed away, certainly deserved the Nobel Prize. So a little bit about her. She worked with a man known as Maurice Wilkins in London. And what they did was they used X-ray crystallography, which was able to capture what DNA is, which is on the right a picture of photo 51, which is the image of DNA. Now, what this did was this provided measurements on the chemistry of DNA. The crystallography is able to look dark in spots where there's a sugar phosphate backbone, and this proved that, this image proved that DNA was in fact 2 strands, not 3 strands that was thrown about by some other scientists. And then that the internal is the nitrogenous bases, part of that nucleotide. So James Watson and Francis Crick, the 2 men who are responsible for receiving the Nobel Prize and for the discovery of the DNA structure. So they put all the pieces together. Watson was visiting Wilkins in London and saw a picture of the X-ray crystallography. So he took that knowledge back and worked on different models. They used Chargaff's rules, knowing that certain nitrogenous bases must be attached to each other like rungs of a ladder. And then also, again, the sugar phosphate molecules would be in the outer strands of the ladder, or what is known as the double helix. So they actually put the pieces together from other scientists, and they earned the Nobel Prize. So what is the structure of DNA? I think we all know that DNA is a double helix. So again, essentially 2 strands put together and wound about, similar to a spiral staircase. Now, the backbone, the sides of the ladder, is that pentose sugar, the deoxyribose sugar, and then a phosphate molecule, PO4. The rungs, or the steps of the ladder, are those various nitrogenous bases. So thymine, adenine, cytosine, or guanine. Now it's important to note that when it's all put together, again, there's 2 sides put together, and they do run anti-parallel to one another, so in opposite directions of one another. It's important to note that the phosphates, which face outward, they have a negative charge and they are attracted to the aqueous or watery surroundings. The nitrogenous bases, which are hydrophobic, are the rungs, and they're tucked inside, not facing the watery or aqueous surroundings. Something to note about the nitrogenous bases: adenine and guanine are also known as purines. So what exactly is that? It's another name for a 2 organic ringed molecule. So again, organic meaning carbon-based. So there's 2 rings for adenine and guanine. Thymine and cytosine, they're not called purines, but they are called pyrimidines. Now, what does that mean? That their molecule is 1 single organic ring. Again, organic being carbon-based. So the pairing as it goes, I think most people can remember this. You always have a purine and a pyrimidine. A's are equal to T's, so they, in the rungs of the ladder, will bond to one another, and G's and C's go together. Now, how I initially learned this, the letter C and the letter G look very similar to one another, and that's how I remember that they go together. Okay, and then try to remember purines and pyrimidines, 1 of each. 2 organic to 1.